Today we're very lucky to have Johanna Rhodes as our invited speaker. She joins us from the Netherlands. So our day is already done. So thank you for doing that at the end of the day. Johanna is doing research on antifungal resistance, which she's going to talk about to us today. But she's also done many other types of work on fungal pathogens in general. Before being a research fellow in the Netherlands, she was a research fellow at Imperial College London, where she did some work on the population genomics of Aspergillus fumigatus and in the environment, and also isolates that infect people. And before that, she was trained at University of Warwick, where she did a undergrad degree, which I didn't know about, in computational biology. So that's where your expertise comes from, in computational genomics. And then she did a master's degree in systems biology and a PhD in systems approach to biological research. And recently, she's had important publications, as I mentioned, on Aspergillus fumigatus, but she also worked on the evolution of ploidy and resistance in Cryptococcus. And she's also worked on epidemiology and genome analysis of Candida auris in the UK. So she's touched many species that many people here care about. So I'm really looking forward to your talk, Joe. And I have to mention also, if you've done some work on antifungal resistance, Johanna was involved in creating the first database on antifungal resistance mutations that was released in 2018, I think. That was very useful for us when we started working on this. And now some of you are working with us on updating the database, and we were in touch early with Johanna to get advice from her on how we should do things, and she's involved with us on this project. Okay, thanks a lot again for being here, Johanna, and please share your screen and go ahead.
Thank you. Right, okay, so what do I want to do? So I feel like this always takes a while to do. So I want to do, nope, here we go. And which one are you seeing? Are you seeing my view or the view that you're meant to be seeing?
We're seeing your view. So if you swap, yeah, perfect now.
Okay, so thank you so much for inviting me to give this talk, and a really super nice introduction as well. Honestly, I've been really looking forward to this. So yeah, I'm going to be talking about evolution of drug resistance in pathogenic fungi. And oh, okay, hi. So if I sort of like look like I'm not talking to you, it's because I'm looking at a different screen as well. But yeah, so fungi are ubiquitous. We know this. They diverge from animals. Fungi and animals diverge from other eukaryotes, shall I say, around 1 billion years ago. And then the fungi and plant kingdoms diverged around 600 million years ago. And there's thought to be at least 150,000 fungal species, but this has been estimated to be higher. So there was this paper by Hawksworth and Looking back in 2019 that put the estimate between 2.2 and 3.8 million. And they estimated this based on this influx of metagenomic data and these so-called dark species. And they thought, there's actually probably more here than what we think. And then there was this paper that came out just last week, I think it was, and they've put a really good estimate of 2.5 million fungal species. So it's quite clear that we're missing quite a bit, really. But from this 150,000 fungal species estimate, we think that over 8,000 species cause disease in plants, and over 600 are associated with humans in terms of causing disease as commensals or pathogens. So we probably all of us know about this, but I just wanted to have a little bit of a refresher. So there's 4 sort of basic types of fungi. You've got the Chytridomycota, which are sort of like these water molds, which are in the top left corner there. And then you've also got zygomycota, which is in the bottom right, things like mucor. You've got the basidiomycota, which are probably what you more typically associate with fungi in terms of these mushrooms. But it's important to note that whilst all mushrooms are fungi, not all fungi can create mushrooms. Then you've also got the ascomycota, such as in that top right panel there, which are the things like neurospora and such like that. And over recent decades, we've seen an increasing number of outbreaks due to fungal diseases. So, for instance, in Europe, we're seeing mass amphibian declines due to fungal pathogens. We're seeing white nose syndrome in bat populations, so they're declining in North America. And what happens here is this white nose fungus covers the face and basically prevents the bats from using their echolocation to search out for food. And we think, well, I say we, the community as a whole thinks that this has potentially been imported from Europe to North America. And then we're also seeing the pathogens that are targeting plants as well. So we're seeing, ash dieback fungus that's targeting, say, chestnut trees, but then also the fungal pathogens that are targeting the important crop species as well. So wheat, rice, maize, things like that. And then ultimately, we're seeing the pathogens that are infecting us as humans. So yes, in recent decades, we're seeing an increase in fungal infections in both plant infecting fungi and then also animal infecting fungi. So the public health threat posed by fungi to us as humans is increasing. And this is because there are new emerging at-risk patient cohorts. So with COVID, we've seen that there are associated fungal infections like Candida auris, Aspergillus fumigatus, and then also the mucormycosis. So when we saw that second wave of COVID going through India, we saw that following that was this wave of mucor infections. We also see at-risk patient cohorts due to drug treatment. The public health threat is also increased by new patterns of drug resistance. And this can be caused by the dual use of antifungals. And by that, I mean you have the same drug class used clinically, but then also environmentally, predominantly in agriculture and horticulture. So pharmaceutical companies, they make antifungals. It costs a heck of a lot of money to produce just a single drug with sort of tens of millions of dollars. And obviously they want to try and maximize their profits, so they introduce this dual use. And what we're seeing now is that there are a few new drugs that are coming through, and we're trying to sort of make people aware that actually they should be for use just clinically or just agriculturally to try and limit the development of drug resistance. Whether that happens doesn't matter. We also see the emergence of highly fit resistant clones, and I'll talk a little bit about that later on. And all of these together are sort of really making the public health threat to humans quite drastic. But fungi, it's important to remember, are opportunistic infections. If we could anthropomorphize them, they don't want to infect us because we're kind of a dead-end host. But they are infecting us, and they are becoming increasingly resistant to the first-line drug of choice, which is the azoles. But we're also seeing resistance to other antifungal drugs, such as the echinocandins, the polines, the pyrimidine analogues, as well. So I'm sure we're all aware, last year, the WHO released this fungal priority pathogen list. And you've got 19 species, groups of fungi in here, ranked as either medium, high, or critical priority. And this is based on a number of things there. The mortality rates, the drug-resistant rates, the global distribution. So for instance, there's coccidioides, which is in the medium priority group down at the bottom. That wasn't a higher priority, mainly because it's limited sort of geographically, whereas something like Aspergillus is more sort of globally dispersed. So the mortality rates in Cryptococcus neoformans, which causes fungal meningitis in HIV/AIDS patients, is anywhere between 41 and 61%. Candida auris in invasive infections the mortality rate is between 29 and 53%. Drug-resistant Aspergillus fumigatus can cause a mortality rate of up to 88%. And then Candida albicans, similar to Candida auris in terms of mortality rates in invasive infections as well. So historically speaking, when we've looked at fungal outbreaks and tried to employ an epidemiological approach, we've had some standard approaches. So firstly, we've really utilised these field surveys and observational studies. So this is Matt Fisher here, who's in Zambia, and he's actually holding in his hand an air sampler, and he's sampling the air to see what fungi are present and then employing this meta barcoding approach. We can also have case control studies to investigate the risk factors associated with disease, cohort studies looking at specific patient populations, outbreak investigations where we're trying to find not only the source, but also how it's being transmitted. Then, of course, you've got the lab-based techniques to try and identify and characterize the fungi, and then the associated data analysis to look at associations, trends, things like that. But then recently, in the last couple of decades, genomics has come along, and that adds different technique to apply to our epidemiological investigations. So the WHO defines genomics as the study of the total or part of the genetic or epigenetic sequence information of organisms. And it's attempting to understand the structure and function of these sequences in the downstream biological products. So this graph is for human genomes, but as you can see, the cost of sequencing the full human genome in the space of a quarter of a century has rapidly decreased. So if it's that cheap for humans, you can understand that it's probably very cheap to sequence a single fungal genome, a single fungal isolate now. And interestingly, I had a quote to sequence some fungal genomes the other day, and it was €60 per isolate, including the library preparation, which was just so cheap. I was just thought, why wouldn't you? And then we can apply genomics to epidemiology to basically determine the distribution and spread of an infectious disease using the genomic data. And we can do this using a number of different technologies. So by far and large, the most commonly used technology is short read, for instance, from Illumina technologies. The reads are short, so perhaps up to 250 base pairs. They are very accurate. They are high throughput, but the machines themselves, it represents a very high upfront cost for the equipment. So what tends to happen is that you have sequencing facilities that have these sequencing machines, and then when you want your stuff sequenced, you're in a queue, which isn't great in an outbreak situation if you've got to wait, a few days, a few weeks to have your stuff on the machine. Also, one of the downsides is scalability. So at the start of outbreaks, for instance, we might only have 1, 2, or 3 isolates. And that can be very expensive to run on an Illumina machine, just 1 or 2 or 3 isolates. We end up with a heck of a lot of data that we don't really need. So it doesn't really scale. These benefit when you're sequencing lots of isolates at one time, because you can multiplex them. What's a little bit better in terms of scalability is using something like the MinION from Oxford Nanopore Technologies, which is portable and has low capital costs as well. However, the reads are long, but they tend to be less accurate than Illumina data. You can also get PacBio do long read sequencing as well, but again, you've got the same problem in terms of the high upfront costs, similar to Illumina. And genomics has really sort of come into its own in terms of epidemiology from the COVID pandemic. So I think pretty much everyone knows what an epidemiologist is now. My parents certainly do. When I told them I wanted to be an epidemiologist when I was a teenager, my parents just sat there and they were like, oh, why do you want to work with skin? Because they were thinking epidermis. And then I have to go, okay, fine. Now they understand what I do. They still think it's to do with viruses though, so we're getting there. But everyone sort of knows what an epidemiologist does now, and I think a lot of the general public understands the value of genomics in terms of identifying that next strain when in COVID, and they recognise perhaps a phylogenetic tree in terms of, when they've looked at it on the news. So this has really helped. But how useful is genomics in mycology? So I'd say that the majority of research is focused on using genomics to find out why and how fungi are pathogenic, and then also why they're drug resistant as well. And we can do this by comparing genomes, and in this way we can identify mutations caused by single nucleotide polymorphisms like SNPs, and these may contribute to drug resistance or other things as well. We can compare genomes to identify when drug-resistant strains are spreading, so within hospitals, but then also on a larger scale in terms of across the globe. And we can study evolution to reveal genes that are involved in host adaptation. There's some fungi that are endemic to regions of the world and cause severe infections. And then we can use genomics to study how these species differ. So, for instance, the pathogen that causes the mass amphibian declines, Cryptococcus, that is endemic to, say, the Far East, and it doesn't cause infection there, but it's been moved to other parts of the globe and it does cause infection. So what is different, or is it something to do with the host as well? Genomics can be used in diagnostics, such as RNA-based methods, surveillance, which I'll talk a little bit later, so looking at their genetic similarities and differences, which can also be used in outbreaks. So genomics can be useful for researching non-pathogenic fungi as well. So Saccharomyces cerevisiae is kind of the model organism for eukaryotes, if not fungi as well. And it's been used in fermentation for a very long time. And genomic sequencing and analysis has shown that there's been 2 separate domestication events, one for sake and another for wine. So these domestication events based on sort of geographic info as well. But there are challenges associated with fungal genomics. So there's over 1,000 fungal species that have been whole genome sequenced, which when you think back to my earlier slides, that's really not a lot compared to how many fungal species we know there are. And it's certainly not a lot when we know that over 130,000 bacterial species have been sequenced. For fungi, the genomes can be quite large. They can range in size from anything from 2 to 180 million. So if you think about something like Malassezia, it's quite a small fungal genome. And then other fungal genomes can be quite large, and therefore their predicted proteomes can be quite big as well. And there is a difficulty in extracting DNA in some cases. So lysis buffer may not degrade the cell wall, in which case you need something called bead beating. And if you've never done this before, you have your Eppendorf with your fungus in it, and you literally have these silica beads, which are about 1 millimeter in diameter, and you put it in a machine that just shakes everything up for about 45 seconds, and that degrades the cell wall. So that's really good in getting the DNA out. But the problem here is that the bead beating probably degrades your DNA as well, which means that it's not great for long read sequencing. Great for Illumina, that's fine, but for long reads, you really want those longer fragments, and then you'd have to have a different approach in how do you extract your DNA. Fungi also have different life cycles as well. So as I mentioned earlier on, fungi, all mushrooms are fungi, but not all fungi produce mushrooms. Mushrooms are in a particular part of the life cycle of a fungus. So do you sequence the DNA when it's in a spore form, when it's in a hyphal form, or when it's in the mushroom form? When is best to extract your DNA? And it probably depends on what questions you're asking. The genomes can be haploid or diploid, or they can even display aneuploidy as well, which I'll talk a little bit about in a moment. And for pathogenic fungi, they certainly display a large number of drug resistance mechanisms. So, aneuploidy. So this is Cryptococcus neoformans here at the bottom panel. It is an encapsulated yeast that causes fungal meningitis in HIV/AIDS patients. And what was really interesting is this study by Edward Sionov, 13 years ago now, he noticed that when you grew Cryptococcus on media containing fluconazole, which is the first line, or was the first line drug of choice, for instance, for treating cryptococcal meningitis, you actually saw a copy number increase in the chromosome that contains the gene ERG11. So this was due to aneuploidy. Most eukaryotic species have two diploid chromosomes, or maybe even more than two sets of chromosomes, therefore polyploid. This might be as a result of ancient whole genome duplication, or maybe even hybridization events. A lot of clinical fungi are actually haploid, they only contain one. But they undergo ploidy changes during adaptation, maybe to adverse or new environments. And then you've got some fungi that actually exist as stable diploid, haploid, polyploid, and then undergo the ploidy changes, and then can even revert back when the pressure has been removed. So if aneuploidy equals an abnormal chromosome number, we can see this in novel environments, periods of stress, and then also during these ploidy level changes. And traditionally, we would detect aneuploidy based on flow cytometry. And I haven't ever done flow cytometry. I try to avoid lab work if I can. But from what I've been told, it wasn't particularly easy. And there were disadvantages with trying to detect the aneuploidy with flow cytometry. So for instance, if an isolate had a single aneuploid chromosome, it might not be significantly different. And isolates with multiple aneuploids may not show different DNA fluorescence because they essentially cancel each other out. So genomics has actually been a huge advantage for detecting aneuploidy, because you can do something as simple and sort of like a quick and dirty normalized coverage of the whole genome, and you can literally see the increases or decreases compared to the normal coverage. So yeah, so if you look on the right-hand side, I can't kind of get my pointer. On the right-hand side, in the middle panel, at the bottom, you've got an aneuploid, and you can see these increases and decreases in chromosome number. So this has been really nice. You can pick up the increase or decreases. You can also pick up segmental chromosome aneuploidies, and then also more fine scale gene duplications and deletions as well. So in Saccharomyces, environmental isolates can include stable haploids, diploids, but also polyploids. It's the clinical and industrial isolates that show the aneuploids. So these are the ones that we use for, fermentation processes, for wine, for beer, for all sorts. And then we can also see this, this stable aneuploidy in sherry and ale strains as well. In Cryptococcus neoformans, we see an amplification of chromosome 1, which houses the ERG11 gene, linked to antifungal drug stress and resistance. In Candida albicans, aneuploidy is seen as adaptation to growth on alternative growth sources, also exposure to drugs, high temperature, and host interaction. So aneuploidy can be induced due to a variety of different things. The second drug resistance mechanism is heteroresistance. So this is something that is kind of common in bacteria. It's a form of antimicrobial resistance where there's a minority subpopulation of resistant cells that coexist with a majority susceptible population. And what tends to happen is the treatment kills the susceptible but not the resistant cells. They survive and they replicate. And then you've got that low frequency of resistant cells that wouldn't have been picked up when you're testing originally, because this relies on liquid-based assays. So in theory, you treat the infection, you think you've got it all, but you've got this really small population of resistant cells. This can mediate treatment failure, but this has mostly been seen in mouse models, and it's less so in humans, and that's because treatment is usually done in combination. So in Candida parapsilosis, treatment is with echinocandin, so it's a different drug class. And you have this prophylaxis exposure linked to an increase in infections. And this paper asked why. Why is that happening? And it's because there was heteroresistance to a particular echinocandin called mycofungin, colonizing the intestinal tract. In Cryptococcus neoformans, we also observed heteroresistance in the cerebral spinal fluid of patients at baseline. So Cryptococcus neoformans, just as a reminder, it causes the fungal meningitis. So taking the CSF from patients is a way to detect the Cryptococcus. And normally, we would treat cryptococcal meningitis with fluconazole monotherapy. And what happened is that the resistant colonies increased, but there were no resistant colonies found when you treated with fluconazole and flucytosine, so combination therapy. And what we saw with whole genome sequencing, there were high rates of aneuploidy in heteroresistant colonies that were basically driving the resistance to fluconazole monotherapy. And this heteroresistance was suppressed with combination therapy. So this paper came out and we kind of described it as putting the final nail in the coffin of fluconazole monotherapy and recommending that actually combination therapy is the way to go. Finally, hypermutation. This is personally my favorite. I love talking about hypermutation because I just think it's really wacky. It's a really commonly studied area in bacteria and less so in fungi. So it's only really been in the last decade that people have started to sort of consider fungal hypermutation. And hypermutators have this elevated mutation rate due to defects caused by non-synonymous SNPs, which encode amino acid changes, in genes in the DNA mismatch repair system. It can also be due to nonsense mutations where they encode a stop codon and cause a premature truncation of the translated protein. So again, in Cryptococcus neoformans, what we saw was that we had 17 patients who had relapse in infection. And there was one particular patient who relapsed after about 180 days. And then when we looked at the whole genome sequences, day 1 compared to day 180, they were phylogenetically dissimilar. So initially we just thought, oh, that's not very nice. You know, that patient has been unlucky. They've been infected twice by something completely genetically dissimilar. And that was because there was large numbers of SNPs, phylogenetically dissimilar, aneuploidy was observed. But what we actually saw when we went a little bit deeper was this, it was actually a relapse because the initial infection contained 2 nonsense mutations in MSH2, one in MSH5 and one in RAD5, which they're all DNA mismatch repair genes. And this resulted in premature stop codons, so the protein was truncated in these 3 genes. And this led to massive amounts of mutations all throughout the genome and then aneuploidy. So basically, when they were treated initially, the drug treatment didn't work because it was drug resistant, and actually it was probably pan-drug class resistant. And then that led to a relapse infection and actually a poor patient outcome as well, should we say. And we think that people are picking up these hypermutators from the environment as well. They're breathing them in and then they're causing this relapse infection. So why does resistance occur in the first place, really? And we think basically the answer is because of us as humans. We are driving it. We're driving it through changing agrochemical use. As I mentioned, there are changing at-risk groups. There's more patient cohorts that are at risk of fungal infection. There's a change in climate. So as the world warms, species distributions will change. Things will move to new places. Change in virulence and change in biota as well. And then we're coming back to this dual usage. So if we're using the same drug classes clinically to treat infections in humans, as well as as fungicides in the environment that can drive resistance. Oh, I was kind of hoping this would play, but it won't. So this is meant to be showing the spores dispersing in the air. Because during the life cycle of fungi, they will produce spores which will go up into the air and they'll distribute. And we decided to set up these Burkhardt air samplers in 2 locations. I am not in this photo because this is the roof of the hospital in one of the hospitals in London. And it was a really low wall, and I don't do height, so I'm kind of, plastered against the wall here. But these Burkhardt air samplers, they sample, they constantly sample air. You have to change it the filter every 7 days. So we set one up in an urban location in London, and then one in a rural location as well. And we expected to see differences, but we were quite surprised by the differences we saw. But what was consistently seen is Aspergillus fumigatus, and that's the picture there at the top right. Aspergillus can grow up to 60 degrees C. It grows in soil and compost, and it sporulates a lot, so it's aerosolized. And there's certain patient cohorts that are at risk of developing aspergillosis, which is the infection caused by Aspergillus fumigatus. So cystic fibrosis. So the UK, for instance, has the 2nd largest cohort of cystic fibrosis patients after the US, and that number is increasing as well. So it's something about 12,000 people in the population of the UK that have cystic fibrosis. We expect the number of people at risk of aspergillosis who have cystic fibrosis to decrease with the new drugs that have been approved, but there's still about 10% of cystic fibrosis people who these drugs don't work for, so it's still going to continue to be a problem. And there's other patient cohorts that are also at risk, notably the severe influenza, the IAPA, and then also the Kappa, the COVID-associated aspergillosis, which we've seen obviously since the pandemic. So the drug resistance mechanisms in Aspergillus are different to the ones that I mentioned earlier. They're centered on CYP51A. So this is the same as ERG11 that I mentioned in Cryptococcus. It's just called a different name. And the resistance functions through structural polymorphism. So these are the amino acid changes, and they interfere with the entry of the azole drug into the hydrophobic access pocket of the enzyme. And these are the things here, like this Y121F and the L98H. You can also have overexpression of the gene, and this is caused by these tandem repeats in the promoter region. So tandem repeat 34 is most often associated with this leucine to histidine shift at codon 98. That's the most common resistance mechanism in Aspergillus fumigatus, closely, well, sort of followed by this TR46 mechanism as well. And both occur widely in the environment as well as in clinical infections. And then rather anecdotally, this hasn't been published, there's also the possibility that duplication events of the gene can also contribute to resistance as well. So Ali was kind of the person who I must give credit to, because I wouldn't actually work on Aspergillus if it weren't for Ali. He came to us as a PhD student. He was working in the NHS in London Monday to Friday and doing his PhD at the weekend. So he lives and breathes fungal research. He loves it. And he came to us saying, let's do some whole genome sequencing. That was 8 years ago. The rest is history. But what he found, looking at cystic fibrosis, isolates from cystic fibrosis patients, was that they had azole resistance in 13% of those isolates, and 27% of those resistant isolates had this TR34 L98H. So this was the first sort of study looking at the rates of azole resistance in the UK at the time. And my PhD student Renard has recently followed up with this by doing this really high throughput phenotyping And she's seen that azole-resistant isolates from cystic fibrosis patients that contain this TR34L98H have a fitness advantage over susceptible isolates, but in certain conditions. And those conditions are acidic pH and this high histamine, high histidine content, which are the conditions that are primarily found in the cystic fibrosis lung. So that kind of gets you thinking, okay, so are these isolates, were they already adapted to those conditions prior to infection, and they've just kind of basically succeeded in the cystic fibrosis lung? Or have they been selected for and developed the adaptation in the patient? Or the third question is, and perhaps this is a little bit controversial, the Aspergillus has modified the environment, its immediate environment in the alveoli, in the lung, to suit it, to make it adapt, to make it survive. What was really interesting is we also saw resistance in the environment, but not quite what we expected. We really expected rural fields to have the highest rates of drug resistance because of agriculturally used fungicides. But that wasn't the case. Rural fields only showed 1% azole resistance and just TR34. So this is over southern England as well. So actually, a decent sample. But it was the urban flower beds that contained the majority of the azole resistance, up to 14%. And you also see more variety. You see the TR34 and then the TR46 as well. And this is because we think people are going to garden centres, they're buying their bags of compost, they're buying their plants, which, might have been treated with azoles. So for instance, here in the Netherlands, tulips are a massive export, and those bulbs are dunked in these huge vats of azoles prior to being exported. So the selection pressure is already there in the horticultural industry. The compost heaps are great for Aspergillus. It gets rotated, it gets turned, and they mix and they develop azole resistance. So this was really the first time where we thought, actually it might be more horticultural driven. And then Jenny came along as part of her PhD. She really utilised this power of social media to embark on this citizen science air sampling. She wanted to look at the rates of azole resistance over the course of the year to see if there was seasonality, but she didn't have the means or the resources or the time to basically cover the entire of the UK. So she basically got people to do it for her and share the results with them, so they were participating in this experiment, and it was a huge success. And what she found was that you're more likely to recover azole-resistant Aspergillus fumigatus in the spring than at any other point of the year. So really demonstrating that there is seasonality. It matters when you sample as well. This has been followed up in Vietnam, and the rates are off the charts. We're seeing at least 80% azole resistance in the environment in Vietnam. So this came to the point where we thought, okay, right, how do we track the changes that are occurring in genomes, and can we pinpoint where and when azole resistance originated, basically? And in order to do this, we had to do quite a broad sampling, so wide spatial and temporal sampling. So we've included over 30 countries, and we've sequenced isolates that date back to 1919. And we've also sequenced the isolates quite deep, so as many samples as possible to capture the diversity that's present. And we've also sequenced these isolates to a high coverage to ensure that the mutations, the SNP balls, are high confidence. We can search for known SNPs that confer drug resistance in CYP51A and then other genes as well. But it also allows us to do exploratory analysis for novel mechanisms such as the pangenome and comparative genomics, which I'm not going to go into. I'm going to focus on what is known. So Zane came to us last year, I think. He is a medical doctor by training and really wanted to get involved in some clinical research. And he was really interested in whether we could pick up signatures of environmental fungicide resistance in our sequences. So he focused on the benzimidazole fungicides, which were brought into use in the 1960s, and they've been that particular fungicide has been registered in many countries on tens of crops, about 70 different crop species, I think. And these particular fungicides, they target the beta-tubulin formation in meiosis. And even though Benomil, this fungicide usage has been phased out, resistance has been reported. So for instance, in the UK, usage has dropped from just over 3 million hectares in 1990 to just over 11,000 hectares in 2020, but we're still seeing resistance. So these fungicides really represent the beginning of the serious resistance problems in fungicides. And here, what Zane's done, he's taken all of our isolates, he's made a phylogeny, and he's plotted the isolates that are resistant in green. And they're resistant due to this F219Y mutation. And as you can see, the majority of the resistant isolates are in this bottom clade. There are a few poking out at the top, but they're in the bottom clade here. And you can see that globally, they're distributed everywhere as well. And then he thought, okay, so what about other fungicides? So the QOIs, similar to the benzimidazoles, are a single site inhibitor, but resistance was uncovered to these fungicides a lot quicker, within 2 years of introduction. And again, we're starting to see phasing out of this due to that. So we focused just on the UK, and the colours are flipped here. So if it's yellow, it's resistant. You can see resistance increasing really rapidly over time, and it's all over the UK. And basically, because of this dual drug usage of the azoles, we are seeing a rapid increase in drug resistance. So this is the same phylogeny as what Zane generated. And the blue isolates are the isolates that are resistant to azoles. And as you can see, they're on exactly the same background as agricultural fungicides. They're on exactly the same background as the agricultural fungicides. You're more likely to find azole drug resistance in this bottom clade, still a few poking out at the top, than in the top. And again, globally distributed and increasing through time. So we've termed this bottom clade clade A for azole, the top clade clade B for boring. So the majority of azole resistance mechanisms are in clade A, and the majority of isolates in clade B are azole susceptible. And we wanted to see if this matched up with the agricultural fungicides, and the short answer is yes. You're more likely to find agricultural resistance in clade A, so this is MBCR, than you are in clade B. But what's really interesting is that you're also finding clinical isolates that are resistant to agricultural fungicides. So if you're taking isolates from a patient that are agricultural fungicide resistant, that means that the only way that patient has acquired that resistant isolate is from the environment, inhalation. And then we're seeing this pan-drug class resistance. What we're also seeing is the capacity of Aspergillus fumigatus to have these clonal genotypes. So this particular clonal genotype, it's just a few hundred SNPs separating these isolates, and they are found in the UK, Western Europe, and then also Southeast Asia as well. They are resistant to azoles. They all contain this TR34 mechanism, and they're also all resistant to agricultural fungicides as well. They're separated by just a few SNPs, and it's got a really strong p-value associated with it. And they are actually, they do actually have a fitness advantage. So fitness experiments have shown that they have an advantage over drug-susceptible Aspergillus isolates. So we really do believe that because its clinical isolates are genetically similar to environmental isolates, that patients are acquiring these infections from the environment. Gillian was working with me this year to basically address whether clade A just has a higher mutation rate than clade B to begin with. And work on this was actually published on bioRxiv yesterday. So what we can see here is that clade A, which is the purple, actually has more SNPs and more indels, so short insertions and deletions, than clade B isolates. And when she looked at azole resistance associated with mismatch repair genes, so thinking back to those hypermutators from Cryptococcus, you can see a higher rate of SNPs in MSH2, MSH4, and MSH5 in clade A isolates, which may actually result in more beneficial mutations, including drug resistance, increased virulence, etc. So changing tack slightly, genomic surveillance of outbreaks. So pretty much simultaneously to the WHO releasing its fungal pathogen priority list, it also released this strategy on global genomic surveillance as well. And basically it was for pathogens with pandemic and epidemic potential. So we all know the importance of fungi and how serious it can be, but we still have a long way to go to convince everyone else. So one of the comments that I've received today is that the fungal community is small, so why would I bother funding your research? But we know that this is a serious problem. And surveillance has really helped, for instance, here in an outbreak in the kakapo. So I'm just going to hit play here. In case you don't know what a kakapo is, it's a parrot. It doesn't fly, and it lives in New Zealand. It's actually that colour green. It's quite incredible. They're critically endangered. And in 2019, there was an outbreak of avian aspergillosis affecting 21 individuals. And the population is only about 200 anyway. So if 9 died, that's actually a significant amount of the population. So during COVID, there was a group of us in New Zealand, and then also me in the UK, and then in California. We're trying to coordinate these meetings to try and work on this. And we managed to sort of get together and whole genome sequence these strains from infected kakapo and their environments in the nests. And we found that the strains only differed by a handful of SNPs. So they were very similar to each other. So we knew that the outbreak was caused by a single strain that hadn't been previously recorded. We didn't see it in any of our 1,000+ isolates from our previous study, which was really incredible. And the value of this study, we realized that actually, again, the answer is us as humans. It was basically brought in on a lorry as part of the logistics for a supermarket, and then just basically found some sort of hook in the local environment, and unfortunately caused the outbreak. So with this in mind, we thought, right, okay, we really need to develop some sort of surveillance platform for outbreaks. So Pathogen Watch is a global platform for genomic surveillance, and up until fairly recently, it's just been for mainly bacterial outbreaks. But I'm really pleased to say that we have now developed this platform for Candida auris outbreaks as well. The beauty of this is that you don't need to have bioinformatics experience. You can sequence your Candida auris isolate or isolates, multiple. You can drag and drop the FASTQ files, and it will tell you, one, that it's definitely Candida auris. It will tell you what drugs it's resistant to. And if you want, it can combine it with other publicly available data. So you can create a collection, you can create a phylogenetic tree, and you can start to explore the possibility of where your isolate has come from and where it's been transmitted from. To give you a background on Candida auris, it's recently emerged. Up until sort of like 20 years ago, no one had even heard of Candida auris, and it was routinely misdiagnosed as a different Candida species. So it's now found on pretty much every single continent apart from Antarctica. There's 4 main clades, and there's been 2 other clades that have been described recently. And if you look at this phylogeny down the bottom here, each clade is very, very different from each other, and they're separated by tens of thousands of SNPs. Clade 2, which was originally described in East Asia, is mainly drug susceptible, but the other clades are drug resistant, and this is due to different mutations in their ERG11 genes and then other genes as well. So my PhD student Hugh, who has a medical background and then he's coming to do some clinical research on Candida auris, has basically taken all of the publicly available data sets of Candida auris, and he's mapped the mutations in certain genes that might be responsible for azole resistance, and they are specific in certain clades. And he's also looked at mutations in additional genes that are capable of causing not just resistance to azoles, but also echinocandins and fluticin as well. So Candida auris is really quite interesting to work with. It's also quite scary because it is able to just rapidly develop this drug resistance to multiple drug classes. Genomics has been really handy for looking at outbreaks of Candida auris, in particular this outbreak in a single London hospital. So this is a teaching hospital. It predominantly deals with diabetic patients. It has quite a big diabetic cohort. And what happened was the first case of Candida auris was picked up in early 2018. So in the UK, Candida auris is reportable. Most patients that are coming in are tested for Candida auris, and then if they have a positive test, that gets sent off to the UK Public Health Authority, and then that patient is then checked up on to see whether they clear the infection. Obviously, they have treatment. And what happened was the outbreak occurred in multiple patients, and it seemed to go on for a long time until sort of end of 2019. And the hospital couldn't figure out why their infection control measures weren't really working. And what happened was when we applied whole genome sequencing to this, we saw that it was because there was more than one introduction. And it was really clear right from the start, there was at least 2 introductions because you had 2 different, very distinct clades. They were all of them were resistant to echinocandins, but in different amounts as well. So when we looked at this on a phylogenetic tree, what we could see was, yes, there was definitely at least 2 introductions. One of clade 3, which originated in South America, and at least one that was a clade 1 introduction. But what was really interesting, you can't really see much on this phylogenetic tree, but actually within clade 1, there were 2 introductions as well. It's just that one was under more control than the other. So we believe that there were 3 introductions to this hospital, and genomics really helped us understand that. What's amazing is that Candida auris acts a little bit like a bacteria in terms of its genome. It's incredibly clonal, which means that we can take a lot of the tools that were developed for bacteria and apply them to Candida auris. And my student Dana here used this tool called Transphilo, which was written to look at transmission events of bacteria. And she could see that not only was Candida auris being transferred between hospitals, but also within the hospital. So between hospital makes complete sense because in the UK, patients go to a main hospital for big treatment or operations, and then they'll go to a tertiary hospital for rehabilitation. And what was happening was that patients were taking Candida auris with them and basically seeding a mini outbreak elsewhere. But the real value of this was the movement within the hospital. And these arrows show the transmission events. And in a lot of the cases, they actually match up with patient movement because a patient doesn't sit just on the ward. They might go down for an X-ray, they might go to the operating theatre, and you can basically see the movements, or they might just even move to a different ward if space is needed. And these movements tracked with the transmission events that were shown here. The final thing I wanted to talk about was a new emerging fungal pathogen. So if Candida auris has taught us anything, it's how to use genomics and apply it to new emerging fungal pathogens to try and figure out what makes it tick. So this is trichophyton. So Trichophyton metagrophytes variant 8, which has been reclassified as Trichophyton indotineae, causes this inflammatory skin disease. It's not particularly nice to look at. It's also not nice to experience. It's not going to kill you, but it's going to make you incredibly uncomfortable. And it's also hard to treat. What's interesting about this is that T. indotineae is a human disease, but other forms of Trichophyton are animal diseases. So it's had this animal to human jump. It's terbinafine, which is a different drug class resistant, and we've started to see first reports of this resistance in 2017 in India. We're now seeing reports in the Netherlands, in France, in the UK, and also in Canada. And the resistance is due to mutations in the SQLE gene. We call it the SQLE gene. So we also believe that this can be transmitted person to person as well. So basically bypassing the animal transmission. So work done by Chloe here. She's basically applied whole genome sequencing to a bunch of isolates from India, France, Netherlands, UK, and Canada, and tried to figure out what makes it tick. So what she found was really interesting, was that certain amino acid substitutions are associated with different levels of drug resistance, so different MICs to terbinafine. So you're more likely to find this leucine, this 393 amino acid substitution at high MICs than you are, say, a different amino acid substitution. And at intermediate MICs, you're more likely to see this 414. And these might be working in combination with other mutations in another gene as well. So we really need to tease this apart and look at perhaps epistatic interactions here. So there's more work to be done on that, but hopefully, in summary, I've shown you that genomics in mycology has a lot of advantages, and there's also been a lot of advances over the last few years, even just in like the last decade. Short read sequencing has improved, but then you've got this long read sequencing becoming more commonplace, and it answers and addresses different questions. Sequencing in general can help our understanding of the evolution of drug resistance in fungi, and when applied to epidemiological studies, can really help us identify transmission events, when and where. It helps with outbreak surveillance. And then as these technologies, but also the software to analyze, continue to advance, more discoveries will be made. So resistance mechanisms are not only SNP-based, and I didn't talk about this, but we're also starting to really recognize that resistance mechanisms might be down to structural variation or the accessory genome in the pangenome. So thank you very much for your attention. I'd like to thank everyone on here, and I'm happy to take any questions that you might have.